Good afternoon, dear students. Lecture number 9. The topic of the lecture, molecular genetic and cellular mechanisms of regeneration, stem cells. Lecture plan. At first, regeneration is one of the main properties of living organisms. Types of regeneration. And the next one, stem cells. Types of stem cells, sources of stem cells, and the use of stem cells in medicine. Regeneration is the ability of living bodies to restore their structures. The term was coined by the French scientist Réaumur in 1712. He studied the restoration of the legs of crayfish. He told about this process as a secondary growth. Regeneration is subdivided. Physiological restoration of body structures that are worn out in the process of life. Reparative restoration of structures after damage. There is no sharp difference between the two types of regeneration. Reparative regeneration is an enhanced physiological one. Regeneration in response to damage can be complete. The structure is restored completely. Incomplete. The structure is not restored completely. Typical. What was lost is restored. It is called homomorphosis. And atypical. The restored structure differs from the original. Heteromorphosis. There are three sources of regeneration in body. At first, dedifferentiated cells. During dedifferentiation in cells, the activity of type-specific gene that determine the cell specialization is suppressed. Then the genes necessary for cell proliferation are activated. And the next one, regional stem cells. Such cells are found in many tissues: muscles, skin, epidermis, liver, retina, and even nerve tissue. It is believed that they are the source from which differentiated cells are formed. As the age increases, the population of such cells decreases. And the next one, stem cells from other structures that have migrated to the site of regeneration. This is observed if the organ lacks its own regional stem cells. It has been shown that stem cells isolated from one adult tissue can give rise to mature cells of other cell lines. For example, the endothelium of large arteries doesn't have its own stem cell reserves. Its renewal is due to bone marrow stem cells entering the bloodstream. Let's speak about stem cells. The end of the 20th century was marked by a number of major discoveries in molecular and cellular technology. One of them was stem cells. In 1999, the journal Science recognized the discovery of embryonic stem cells as the third most important event in biology, after the decoding of the DNA double helix and the Human Genome Program. The founder of science of stem cells is the professor of the Imperial Military Medical Academy, Maximov. The fundamentals of the science of bone marrow stem cells were led by Alexander Yakovlevich Wittgenstein and Joseph Lvovich Chertkov. In the 1918s, Thomas performed the first bone marrow transplantation in a patient with lymphoma. In 1992, David Harris first collected and preserved the umbilical cord blood. In 2001, in the USA, they performed the first successful transplantation of neural stem cells to a person after a stroke. Transplantation of autologous skeletal myoblasts to a patient with myocardial infarction. What are the stem cells? Stem cells are the populations of immature cells with unlimited proliferation and capable of differentiating into various types of cells. Stem cells are characterized by the following properties. The ability to unlimited division, self-renewal. The next one, determination. And the next one, complete differentiation. There are several types of stem cells. They are on the screen. Totipotent stem cell. The zygote and blastomeres, or the first few cleavage divisions, is capable of producing the whole organism. Pluripotent, omnipotent stem cells of the blastocyst embryoblast cells are capable of producing all types of tissues. And multipotent blast stem cells, immature tissue cells, osteoblasts, myoblasts, and etc. Unipotent cells of body tissues give only one type of cells. Independence of sources, there are following stem cell types: embryonic, fetal, somatic, mesenchymal. Well, let's study all these types. At first, embryonic stem cells. These cells are found at the earliest stage of embryonic development. In our country, one of the recognized leaders in the field of embryonic stem cells is Professor Repin. The most important property of embryonic stem cells are: the genetic information contained in the nucleus is at the 0 point of reference. The genome has not yet launched any program and has not begun the program of the reproduction and formation of a multicellular embryo. Islets are formed from embryonic stem cells in various organs and tissues. Features of embryonic stem cells are: totipotency, the ability to form any of about 350 types of cells in the body. Homing is the ability of stem cells, when introduced into the body, to find the zone of damage and fix there, performing the lost function. Telomerase activity. With each replication, a part of the telomeres is lost. In stem, germ, and tumor cells, the ends of chromosomes are kept long. That is, these cells are able to undergo a potentially infinity number of cell divisions. They are immortal. The next type of stem cells are fetal stem cells. Fetal stem cells are stem cells from which various organs of embryo develop. Nowadays, well-studied neural, homopoietic, pancreatic, progenitor cells. Somatic stem cells. They are able to transform only into certain types of cells that form the tissues of an adult organism. The sources of somatic stem cells in an adult are bone marrow, peripheral blood, adipose tissue, skeletal muscles, tooth pulp, liver, skin, pancreas, and etc. Cells of this type support tissue renewal throughout life. Mesenchymal stem cells are contained in the adult bone marrow, can profile have a fibroblastic type in culture and form a monolayer, and also have the ability to differentiate into tissues of mesenchymal origin, such as bone, fat, tendons, muscle, and stroma. They show the ability to differentiate into neural and glial direction. These type of cells are actively used in cardiology for cell therapy of various pathologies. The next point of the lecture is obtaining and using of stem cells. Stem cells can be obtained from various organs and tissues: bone marrow, fat, muscle tissue, skin. Bone marrow donors should match recipients as if they are genetic twins. Donors registers of HLA-typed bone marrow donors have been created in Europe and the USA. It became possible to isolate embryonic stem cells from blastocysts and cultivate them in laboratory conditions. Potential uses of stem cells. As you see on the screen, there are a lot of diseases which can be cured with the help of stem cells. So, the use of stem cells in medicine. Many scientists argue that they should be used very carefully. Certainly, stem cells can be used for the treatment of blood cancers, breast cancer, neuroblastoma, as well as for the treatment of thalassemia, sickle cell anemia, Fanconi anemia, systemic lupus erythematosus, multiple sclerosis, Parkinson's disease, Alzheimer's disease, and etc. Thus, despite the ambiguity of the opinion of specialists regarding the stem cells, great hopes are pinned on their use. But there are pros and cons of cell technologies. Cons are stem cells can be obtained from a fetus of 9 to 12 weeks of intrauterine development. A check of the source of stem cells for infection with herpes viruses, hepatitis, human immunodeficiency, and other dangerous infections is necessary. Society as a whole misunderstands such experiments in the field of biotechnology. Pros are the successful use of stem cells will help in the treatment of diseases such as cancer, coronary heart disease, metabolic diseases, anemia, and many, many others. Thank you for your attention.